All right,
You know,
so
I always
we are hooked up on Facebook, so I'm going to go ahead and give you a quick introduction. So
Okay.
good morning, everyone. Welcome back to Ask a Scientist with the Asheville Museum of Science. My name is Abby. I'm one of the educators with the museum. And today we have Dr. Tanisha Williams with us. So Dr. Williams is a plant lover and expert. And over the years, her research has taken her to places such as Peru and South Africa. So some really biodiverse areas. And she's looked at everything from plant genetics to species distribution, climate change. Her work has earned her the title of a Fulbright Alumni Ambassador and led her to participate in congressional visit days in Washington, D.C., so she can make educated decisions with people about botanical research. So she's now a postdoctoral researcher. So she got her PhD and said, but wait, there's more. And she is doing a number of really interesting projects right now that she's going to talk with us about, including the role of indigenous people that they have on some plant ecology in Northern Australia. So get ready and send in some questions, and Dr. Williams will get us started.
Thank you so much, Abigail, for inviting me here. And I'm super excited to talk to all of you. I just want to preface now that my internet may be a little wonky. We're having a very windy day here in Pennsylvania. And if that happens, I may turn my video off, but you'll still hear my voice. But I'm going to keep it on for right now. So today I'm going to be talking about conserving biodiversity from South Africa to Australia and some other places in between. It's going to be a whirlwind tour. So let's start from the beginning. I'm going to tell us a little bit of a sad story, and then we're going to bring it up and lift our spirits towards the end of the talk. But I'm sure that all of you guys know this same old story, that climate change is one of the greatest threats to life as we know it. We're seeing these increasing in temperatures above pre-industrial levels, coupled with greenhouse gases increasing and sea level rising. And these all lead to events such as increased heat waves and drought events, intensities of storms that we're hearing in the news. And that all impacts our food security and water availability and things that we don't even know right now. A colleague of mine at the University of California, he wanted to look at extinction risk, and he calculated that by 2100, one out of every species will become extinct. And we're seeing a lot of that pressure in South America in red, in Africa and Australia. So these are alarming statistics. And I am a botanist and a plant ecologist. I'm really jazzed about plants. And so I think about these extinction risks in plants. And so based on this Kew Gardens study, more than 50,000 plant species have been estimated to be near extinction. And out of that report, this was a quote that really just really grabbed my heart, where I'll read this last part. It says, "There is widespread recognition that plants may become extinct before they have been recognized as being at risk, or perhaps before they've even been discovered." So we're still discovering plants and animals, many plants and animals to this day. And we don't know, not only for discovery's sake of knowing what's on Earth, but we also don't know if these plants have different medicinal values or cures to different disease and ailments that we have. So this is another cause to be concerned and want to conserve biodiversity that we have here on Earth. Based on another study that came out in 2019, Humphreys et al. Showed that the IUCN Red List of extinctions, they're calculating that we're seeing more extinctions than are on the Red List. So the Red List is very strict. The species in the wild has to be extinct for a number of years, and it's a really long time, where Humphreys et al. Says, like, we have experts that are on the ground. They can tell you that it's extinct, it won't grow back, and we don't have to wait a decade to let you know that this plant is extinct in the wild. And they calculated that we're seeing about 2 species per year extinction, which is a really high rate based on previous extinction risks calculated from millions of years ago. In that area, somewhere where is near and dear to my heart, where I worked, is the Cape Floristic region, and I'll tell you more about that soon. This is ranked as the world's second worst area for plant extinctions. We're seeing extinction of 40 species since the 1990s. If we were in person, I would ask you this question. I would ask you, so what is the first place in the world that has the highest number of plant extinctions? And I've gotten so many diverse and great answers, but the answer is Hawaii. So Hawaii is number one, and the Cape Floristic region of South Africa is number two. And on top of having all of those, species extinction, the South Africa in 2017, all the way until about 2018, 2019, was in the news nationally and internationally for their Cape Town water crisis. So they saw the worst drought that they've seen in over 100 years. And this is just a graph of what one of the water towers in the Cape Town region, and this looking at how the water levels are dropped and dropped and dropped since about 2015. And we do see an uptick in 2018. And 2019 was an okay year, but we're still seeing these really low levels in rainfall throughout that region. So droughts are really prominent in that region. And what I have here on the bottom is that Cape Town never reached this point, but they were very, very close to turning off the tap water. So it was going to be the first major city in the world to have all tap water off in homes, off in businesses, besides like hospitals and things like that. So this was really big during my research. And actually the Yukon South African group, we didn't go to South Africa 1 year because of this, because with plants and with what we were working on, we use a lot of water. So we were like, we're just not going to stress the system out. We just won't go this year. So after I told you all that doom and gloom, so how might plants respond to climate change and all of these impacts that we're seeing? I like to think about it in three ways that plants may respond: using phenotypic plasticity, adaptation, or migration. And they can use two of these, three of these, a combination of these strategies to survive. So let's break down these strategies. So phenotypic plasticity, I like to think about it, let's say an example of leaves changing. So here we have on this axis, this leaf thickness. So low leaf thickness, think of like a house plant for the most part has low leaf thickness, and high leaf thickness we would think of like a succulent or a cactus. And right here on this axis, we're looking at a wet or dry environment. So in general, we project that in a wet environment, leaves are going to have low leaf thickness because they don't have to worry about desiccation or drying out. Where in a dry environment where they do have to worry about that, they're going to have really thick leaves, and that's going to help them conserve water. So this is a strategy plants can use if the environment changes. Another strategy is adaptation. So this is the National Phenology Network. And as we're showing here is that plants are flowering about almost a month earlier. So 20 to 10 days earlier than they did. And they're shifting their phenology because the climate is warming up. And then lastly, we're looking at migration. So this is normally what we hear about when we talk about how plants may respond to climate change. We normally think about amphibians. There's no cool map like this for plants, but plants are doing a similar migration shift, moving north. And part of my research is really to understand how will plants respond and can they adapt to this rapid rate of contemporary, so current day climate change. And I use a timeline approach, looking at the past, using herbarium records to assess the flowering phenology. In the present, I do a number of garden experiments, and I put plants in areas that, in environments they're projected to see in 30 to 50 years. And I assess, do they actually use phenotypic plasticity? Does that morphology change? And then in the future, I use species distribution models to see how habitats will change and are the species likely to shift to these habitats? So I'm going to introduce us to South Africa, and then I'll tell us a story about that flowering phenology. So South Africa, after COVID is done, everyone should go to South Africa. It's the third most biologically diverse country in the world. It's the fifth highest in the number of plant species. So as a botanist, I'm jumping for joy to visit South Africa. And this fifth highest number of plant species is because the high level of endemism, meaning that these plants are found nowhere else in the world. And as we see in this map here, South Africa has 9 biomes. So it's really diverse, and it's diverse in the vegetation. It's diverse in the rainfall regimes that it has and the temperature regimes that it has. So that makes it a nice place to have all of these different types of plants. I work in a biodiversity hotspot known as the Cape Floristic region. And a biodiversity hotspot is just an area that scientists have called, because of that high level of endemism, they have many, many plant species in a small area, and that area is being threatened by climate change and invasive species and things like that. The Cape Floristic region is the smallest of the 6 floral kingdoms. It's about the size of Pennsylvania, and I think that's pretty small, but I also lived in California, so what's small? And in that really small landmass in the Cape Floristic region, we have about 10% of the world's plants. It's also diverse in reptiles, birds, and even the marine systems are super diverse. So it's a really cool place to work. And it's also recognized by the United Nations, the UNESCO, as a World Heritage Site because of history, culture, and the flora and fauna there. And lastly, it has extensive long-term phenological collections, which I'm going to tell you a lot about. So I work in the genus Pelargonium, and I think most of you guys have seen a Pelargonium because the common geranium that is sold at your local grocery stores or hardwood stores is actually a Pelargonium variety. And pelargoniums are used in many different instances. So it's used in traditional medicines. It's used in modern medicine. So this pelargonium cold relief that I'm picturing right here, you can buy that off of Amazon. And a lot of things, over 80% of the rose scented things are actually coming from pelargoniums, the oils in the leaves from pelargoniums. And they have plants that smell like all sorts of things, from roses, citronellum, the candles, that's coming from pelargonium. So it's a really cool group of plants. It's a highly diverse genus. It has many different types of growth forms, so perennials and shrubs. Subshrubs are just a little shorter than your normal shrub. And geophytes. Geophytes are plants that have tubers, so think about like an onion or something like that growing underground. It's also known for its leaf-shaped diversity, and most importantly, most people love pelargoniums and geraniums because of their beautiful flowers. There's about 280 species throughout South Africa and a couple other countries in Southern Africa, on Madagascar and a couple other remote islands. Out of those 280 species, there's 160 in that small biodiversity hotspot that I showed you. And of those 160, 89% of those are found nowhere else in the world, but in that Cape Floristic region. So what I want to talk to you about is, how has the genus Pelargonium responded to climate change over the past century? And this map is a word cloud of my dissertation chapter. And so we're going to talk a lot about pelargoniums, herbarium records, climate change, and temperature. I always like to stop and really highlight the reason why I was able to do this work is because of many, many scientists and also just citizens collecting plants specimens throughout the year. So I had access to records from 1800 all the way up into 2009. So people love going to South Africa and for its biodiversity, and I was able to use those records. So across the world, we have about 3.5 billion specimens of plants, animals, and fossils. We use them for preservation, research, and outreach, and that's what we use them, those specimens for at the University of Connecticut, where I was. And at Bucknell, where I am now, we're 1 in 3 in the state, and we have about 20,000 specimens here. I like to think of herbaria as this untapped, rich resource, and it's rich in data. Collectors have been collecting plants and animals and fossils for hundreds of years. And we can use this type of data, these collections, to assess pollinator shifts, distribution shifts, phenotypic plasticity. So there's a lot of research we can do with this kind of there's a paper saying old data, new tricks. So I want to make sure we're on the right page about when I'm speaking about phenology. And phenology is just the study of plant and animal life cycles that are cued into environmental events such as temperature and precipitation. And there is a plethora of literature of people using these herbarium specimens to see if plants are responding to changes mainly in temperature, sometimes precipitation. So I'm going to go through a couple of those. So in Ohio, they assessed the temperature record since the late 1800s and saw about a degree change in temperature, although one county, they saw a 2 degrees increase in temperatures. And associated with that, they saw that plants were flowering about 2.5 days earlier. And they had across the 141 species, that's a lot, they saw this not only advancement, some plants were delaying their flowering by 2 weeks, and other plants were advancing their flowering earlier, about a week earlier. And 46% of those show significant advancement in flowering. So it was significant that they were flowering earlier. In Massachusetts, which was close to where I was at the University of Connecticut, we saw a 1.5 degree change in temperature since the late 1800s. And associated with that, we saw that plants were flowering 8 days earlier. And they also calculated out, which I really liked, is that for every degree increase in mean spring temperature, plants were responding to that by flowering 4 days earlier. So what are the trends we're normally finding across the literature? We're seeing that these shifts in flowering phenology in response to increases in temperatures really normally give you an early spring season, which leads to that earlier flowering time. I am from Washington, D.C., so I always like to bring this up. The cherry blossoms are a big part of the city and bring in a lot of tourists. And we can see in a short period of time that the cherry blossoms are actually responding to increases in temperature. And they're flowering earlier, which is something that is very, very something we have to keep track of because we have the parade and people coming. So we have to let them know when to come. Most of these studies are done in North America, Europe, and Asia. And so I wanted to see, do we see the same types of responses in the Southern Hemisphere and also in Africa? So I had access to about 20,000 records from the University of Cape Town and other institutions and databases. And this is what a herbarium record looks like. And this is what I'm looking for. So I'm looking to make sure that record is collected with flowers. And I can say that Pelargoniums, sometimes it's a little hard to find them without flowers. So like 90% of the time, you're gonna find a Pelargonium record sheet with flowers. I also want to make sure I had the correct data. So meaning I want to make sure I had a correct date, a correct species name, and then also a locality that sometimes it was lat long and other times I can geo-reference. So here, this Pikerberg farm, it's the only farm that's named that, and talking to collaborators, I knew where this GPS locality was. And then, it's funny, I have 2 cats and they may take down this Christmas tree soon. So I cleaned the herbarium data, and out of all of those 20,000 records, and we had about 230 species, I narrowed it down to 129 species. and we have collections mean of about 87 collections per species, upward to almost 600 collections per species. Then from there, I had access to environmental data, and that's 2,000 weather stations throughout the Cape Floristic region. So I also pared down my herbarium collections to match that environmental data, since I didn't have the environmental data for the entirety of the country. So the first thing I did was look at how has temperature changed over the past century using this mathematical model. And then if I saw that temperature were changing, I wanted to see are changes in flowers associated with these temperatures using this mathematical model, looking at shifts in flowering time. So getting right in, we can see that for sure, there's an increase in mean annual temperatures throughout the past century. And we're seeing this really huge uptick since the 1980s. I get a lot of questions about that, and I have no idea why we're seeing this fast increase in temperature. So I presented this work to meetings in South Africa, and we're still scratching our heads trying to figure out what that is. So this amount of temperature increase is a 2.9 degrees Celsius increase in temperature, which is, it's getting hot, and it's getting hot fast. Associated with this, we see that that flowering times are also changing. So this Julian date is when the flowering times, when they were collected in flower, and we're seeing that these flowering times are definitely decreasing, meaning that they're flowering earlier. So over the past century, pelargonium flowers are flowering about a week earlier. And we also calculated out that for every increase in mean annual temperature, we're seeing about a 2.5 day advancement in flowering. So for sure, climate change is altering the way plants function and interact with their environment. Long-term flowering records like the records from the herbaria have provided credible evidence and continue to provide this evidence that species are indeed responding to climate change. And that temperature increase in South Africa is quite scary. So it's increasing at a much faster rate than the global rate. So the IPCC panel has calculated this global rate, and we're almost hitting that 1 degree Celsius mark, but in the same amount of time, South Africa is almost hitting a 3 degrees change in temperatures. So this responsiveness to temperatures and flowering makes me, I like to think of my pelargoniums as a canary in the coal mine. So if we continue to monitor these species, they could let us know what's happening at different populations. And maybe this could give us a firsthand response to whether we should be conserving certain sites. So now that we've gone to South Africa, I'm going to take us to Pennsylvania first, and then I'm going to take us to Australia and just give you just a quick tour of what I'm currently doing. So in the lab, I'm working with Dr. Chris Martin here at Bucknell University, and we have a number of different projects that we're working on. And this first one is a project that I'm really excited about, is looking at plant-people interactions. So the Martu people, which is an indigenous group in the Northern Territory of Australia, uses this Solanum diversiflorum plant. It's Solanum is the genus that has tomatoes and eggplants and peppers. So they use this plant on their spiritual walks, and it's a source of nutrients, a source of water. And we're trying to see how these indigenous people, their use of the plant impacts the plant's ecology and genetics. So that's one project that I'm working on. The next one that was started by 2 undergraduate students pictured here, Ariel and John, were describing a new tomato species in Australia currently. So we have it growing in our greenhouse here, and we've taken a lot of measurements and writing up the description. Next, which another aspect that I really love about working here is making sure that our science is actually being used by policy. So we have a partnership with the Western Pennsylvania Conservancy in the Pennsylvania Natural Heritage Program, where we're using our expertise in plant genetics to update the conservation status of rare plants in Pennsylvania. So The first plant in purple, as you go from the right to the left, this is Cheyenne Moore, a master's student. She worked on Baptisia. Then Cheyenne and also the previous postdoc, Dr. Angela McDonald, worked on Aerugenia. And now John, undergraduate student, and I are working on Casmantium latifolium, which is a grass here commonly known as river oats. And lastly, another bringing us to Australia again is we're trying to understand how this legume, Templetonia hookeri, how these biogeographic barriers found throughout Australia are impacting the genetics. And this work is done in collaboration with the old postdoc and Ariel. So I'm going to take us on a little trip through Australia to talk about this project, and then I'll be ready for questions. So Australia is known for its many endemic plants and animals. This island continent is really well known for the endemic plants and animals. It's about 70% of the flora and fauna have yet to be discovered and or described, which is out of this lab, we've described a number of new Solanum species. And so it makes it a really good place to understand species radiations and distribution patterns across the continent. We work in Northern Australia in the Australian monsoon tropics. This is a biome that encompasses nearly a third of the continent, and it's known for its really wet and really dry seasons, but it supports a range of habitats. Now, those biogeographic barriers that I mentioned are these barriers where we normally don't see species gene flow. We normally see this pattern of species disjunction. And that means what I'm going to show here with the common tree fog in Australia, we see the same genus here, but the species is different. So I'm saying species A, B, C, and D. So we're not seeing any gene flow across these white barriers here. I'm going to focus in on two of the major barriers, this Daly River barrier, and then this flanking region. So that dotted line is between the Carpentaria Basin here, and then also this Arnhem Land Plateau here. So we're looking at this barrier here. And we're looking in the species Templetonia hookeri. It's a bean, part of the bean family, and it's associated with these sandstone outcrops. But most importantly, it defies that common biogeographic pattern. So across the barriers, Templetonia hookeri looks the same. So we want to understand, why is Templetonia hookeri so different? So we did some sampling across the barriers, across the Northern Territory. We always like to highlight our collaborators at the Northern Territory, Peter Jabo. This couldn't have been done without him. We extracted the DNA. Then I did a lot of data analysis using a number of different packages. And I always like to highlight that Dr. Angela McDonald, the previous postdoc, optimized this pipeline. I went to a population and speciation genomics workshop in the Czech Republic and further optimized this pipeline that we're using for our Australia work and also our Pennsylvania work. So what we were ended with was about 2 million reads. So this is using genomics data. So we're getting our A, T, C's, and G's. So 2 million of those reads per sample, which is really high quality data. So we had tons of data to work with. So the first thing we wanted to know, to assess, to see if these populations are the same or different, was FST. So this is a measure of genetic isolation. So our overall FST between all population is above this 0.15 threshold that I have here. And that means that populations are genetically isolated. They're not sharing any genetic material. Then we wanted to do a pairwise FST to compare populations to one another. And although we see some of these lighter colors, what I can tell you is that all of these colors are above that 0.15 threshold. So the redder you are, the more genetically isolated you are. So there's very little gene flow going on between these populations. Then we looked at heterozygosity. So that tells us something about genetic diversity. Normally, if we're seeing mixing of populations and different genetic materials, you would see high heterozygosity. And what we're showing here is that all of our p-values are significant, and all of our observed heterozygosity is much lower than expected. So we're seeing very low genetic diversity within these populations. Then we looked at inbreeding. This is also another metric to let us know how the populations are structured. And our overall inbreeding is positive, and positive means there is inbreeding that's occurring. And inbreeding means that there's no outsource of new material. The populations are reproducing with individuals within their populations and not from without. So inbreeding is prevalent throughout these populations. There are two populations, Kakadu and lemon, that do have negative values. So there is evidence that these two populations may be seeing some outbreeding. Then we did a principal components analysis, and this is to look at genetic relatedness. And what I can tell you is that these populations are definitely structured by geography. And let's look at a map. So Victoria and Keep River are structuring together, that Kakadu and Nipmuc are structuring together, and then all of our southern populations are structured together. And these are really structured by those barriers. So in general, what does all this mean? What can we say about that? We can say we saw that FST was low, so very little gene flow. We also saw that there was very little genetic diversity within each of these populations. Inbreeding was prevalent, and the PCA analysis and structure, which I didn't show, but they're strongly structured by geography, and they definitely follow those biogeographic barriers that I pointed out on the map. So populations that are to the left or right of those barriers, they're clustering together based on those barriers. So this is telling us that although Templetonian hookerai morphologically across those barriers looks the same, they are genetically distinct populations. And it's really telling us that radiations within the plant lineages in the Australian monsoon tropics are ongoing. So who knows, thousands to millions of years from now, those Templetonian populations probably won't look the same. So we're doing this type of work in other genera throughout Australia. So we're working on Solanum, Grevillea. Actually, I have analysis running right now in the background, and Boronia. And this was done by a huge team of people. I do like to highlight here, I'm working with Dr. Chris Martin, that's my boss. And I just love the Bucknell environment here because I get to work with so many wonderful collaborators, but undergraduate students. All of these pictures are of undergraduate students. And that's the end of my talk. I just want to point out two more things before I'm quiet and I'll get to hear your questions. So I am the founder and one of the founding committee members of Black Botanist Week. This was a social media campaign held online to highlight Black people who love plants. And I just want to encourage you to follow us on Twitter and check out our website because we're continuing on the work that we're doing. And we're actually partnering up with the Holden Forest and Gardens. And we have a series that every month, each one of the committee members gives a scientific lecture. We've had a lecture on the first Black settlement in New Mexico and how they use dry farming. We've had a lecture on how to navigate a biology career while Black. And the next up is February 10th. Brandy Cannon, who's a middle school science teacher, she's going to be talking about identification and discovery, botanical love story. So please check out the website and join us on February 10th. And lastly, I want to highlight Dr. Chris Martin, who I work with. He is the founder and the spokesperson and everything for this really cool YouTube series called Plants Are Cool Too. And I'm going to play a short clip of the new one, and you can just look up Plants Are Cool Too, and the YouTube channel will come right up. But this is hot off the press. This episode just dropped yesterday.
When most people think of Hawaii, they probably imagine beautiful beaches like this one. But if we want to see the coolest and rarest plants on the archipelago and meet the scientists who study and protect them, we got to get up beyond these sea cliffs and trek into the hills and valleys of the interior. So that's where we're going on the next Plants Are Cool Too. Plants are People from all over the world visit the garden island of Kauai to take in the incredible vistas of Waimea Canyon, often called the Grand Canyon of the Pacific. It's a geologic wonder like nothing else in Hawaii, but it's also among the hottest of the biodiversity hotspots on this string of volcanic islands, with scores of species occurring here and nowhere else on Earth. What are the
All right, that's a taster for you guys. I don't want to play too long, but that is what I have for you. I'll put my contact information up once it figures itself out. And just thank you.
Thank you. That was fantastic. So we do have a couple questions for you, and I can shoot some of them off. We'll start with, what kind of things cause phenotypic plasticity in plants? Is it all about genetic differences, or do external things cause plasticity in plants as well?
Ooh, that's a great question. Yes, so I would say it's not only all about the genetics, although one could say that if you're changing your morphology, it probably has some genetic underpinning, but we definitely look at just morphology alone. So in my work, in my PhD work, I didn't do any genetics work. I only looked at how morphology was changing. So what the outside of the plant looks like. But for sure, it's going to have some genetic underpinning.
And what are some of the threats to biodiversity in Pennsylvania?
That's a good question. So what we're finding out about these rare species is that they're definitely climate change, increases in temperature. So, I mean, what was it? Last winter, there was barely a winter that we had here, rarely any snow. As far as like I'm in central Pennsylvania. We did have snow throughout Pennsylvania, but central Pennsylvania was really, really warm, meaning that a lot of the pests and things like that normally die off during the wintertime were here throughout the entire year. So climate change, I would say the Natural Heritage Program are probably going to say development and invasive species are number one for threats in Pennsylvania.
And you mentioned early flowering and early blooming as a plant response to climate change. Are they blooming longer in the season, or are they dying earlier?
It depends on the species. A lot of species are just have these extended flowering times. I know Pelargonium has that as well, but it depends. It's species dependent. Yep. Good question. These are all great questions.
And have your labs noticed any types of plants that are extra resilient or extra susceptible to these environmental changes?
Ooh, so yes. So during my for my species distribution modeling, I identified 4 species that it looked like their impacts. We were going to see major habitat loss by 2050, and I made projections out to 2070, and their projections didn't even make it that far. They were like done at 2050. So we do see some species that are going to be slam hard, and then there are other species that for sure are resilient, and they may even like find additional populations and additional habitats to move into.
What sorts of advice would you give to undergraduates who are interested in the botany field?
Yes, I'm so excited. If you're interested, I would definitely say if there's a professor at your university that studies plants, if you can get some research experience. I know that during the summer times, even now during COVID, the National Park Service, National US Forest Service, and things like that, they were still providing internships, museums. So definitely looking up internships and museum opportunities. Also, if you're really interested, field guides, you can rent those from your library. so you don't have to buy them, and just going out and trying to key out plants. So using a guide that will help you walk through the different characteristics of leaf shapes and buds and stuff like that, and starting to identify plants in your area.
What was it that got you hooked on plants?
That's a good question. I would say my great grandmother. So I was raised by my great grandmother in Washington, D.C., and I didn't think I was going to be a botanist, but plants were always around me. So throughout my home, we had these wonderful collection of African violets that I would always touch their leaves because they're so fuzzy. Our family went hiking a lot. I was a Girl Scout. We went camping and hiking. So plants were always around.
And so you mentioned to me before we started streaming this that you haven't started too much about your project in Australia with the indigenous groups there, and how they affect some of the plant ecology. But what sorts of things do you think we can learn from indigenous peoples when it comes to conservation, or even preservation?
Oh, that's such a good question. I just want to first say that all of the science that we have thus far have been, for the most part, we're learning from indigenous people, right? When we hear about these discoveries and people sailing across the world, they had to talk to indigenous people to find these plants and things like that. So namely, we're for sure learning so much from indigenous groups, but they have been the caretakers of Earth and of these plants and systems for so long, and they've watched these plants grow. So I think that listening to them and making sure you collaborate with them, that's something we can learn a lot, because they have the knowledge. They have the, as they say, boots on the ground, right? And that's something that I really treasure about my Yukon work, is that we weren't just this American group of scientists flying into South Africa doing this work. Everything I've done has been in collaboration with South African scientists, South African conservation managers and workers. And the same thing in Australia. We're collaborating with universities and herbaria and indigenous groups to make sure we're making sure that the knowledge that we're gaining, it is the people whose countries we're in, it's their knowledge, and making sure we're giving back.
Can you describe the basic process for describing a newly discovered species like that tomato plant you mentioned? How do you choose the name?
Yes. So the first thing is Dr. Chris Martin. He's been going to Australia for 10, 20 years. Well, not 20, but a decade. And so he can, just having his knowledge, can see, like, it's like, oh man, this plant looks so weird. I've never seen it before. Then you look through that botanical key, you don't see anything that looks like it. So he collected it and said, okay, let's grow it up and kind of make sure it's different. And he's like, yeah, this is definitely something I've never seen, as collaborators. So the first thing you do is measure all the traits, meaning we measured like leaf length, different floral characteristics, did all those measurements. And to pick a name, we still haven't picked a name yet. So our lab has done many different, has chosen many different avenues to pick a name. There was a blood red tomato species that Chris went to a local middle school here in Pennsylvania, and had the middle school students pick the name. Right now, I think our lab group is going to pick the name, but some people name it after an important botanist or an important discoverer. Some name it after characteristics of the plant. So on the stem of the plant, we can see all of these we call them prickles, but you can think of them as thorns as well. And so we're trying to see if we can name it something that highlights those prickles along the stem.
If you could name a plant, what would you name a plant?
Ah, that's a good one. I think I would name a plant after my great-grandmother, my late great-grandmother. Her name was Grace. So I would name something like Gracie Eye. I've heard of similar types of plant names. So yeah.
And will you briefly go into how your study abroad helped you in your science career?
Yes. undergraduate students, study abroad if you can. Yes, please look into study abroad. And they do have scholarships. So that's how I was able to go. I was a lot of study abroad programs you do have to pay for, and I couldn't afford that, but there was a scholarship associated with this study abroad program. I will say that just the study abroad program that I was a part of, I got to meet scientists that were researchers working on projects and hear about their research and their passion, also meet the people, the indigenous groups in the rainforest that were working on these projects as well. It just really blew my mind, and something I hadn't seen prior to this. Although I went to Penn State, there's a lot of research going on, but as an undergraduate student, I actually didn't do any research until my master's program. So seeing that in the study abroad program and how passionate people could be about plants and the environment, that really just opened my mind. And Peru, I mean, there's so many cool plants.
I know, I had actually no idea that it's like South Africa was so biodiverse, because whenever people talk about that, especially with plants, they always talk about the rainforest and the Amazon in South America. And so that's really interesting.
There are so many other places in the world that are just as biodiverse or even more. What we call South Africa, we call it the knee-high rainforest because it doesn't get as much water and rain as the Amazonian rainforest. So you're not going to get these huge, big trees. But if you look from the knee down, there are so many plant species. It's really, really diverse.
And when you talk about outbreeding in plants, is that the first step of plant evolution?
Yeah, for sure. For sure. That's one of the first steps. That's a good question. Yeah.
All right. And we are coming down. I don't think we don't have any more coming in right now, but do you have any last minute thoughts to leave us with?
Yeah, I just want to encourage everyone, if you're interested in plants and the environment, we need more of you. If you watch that Plants Are Cool episode, Chris at the end basically pronounces this like call to action. He's like, we need more botanists that are trying to save the world. We for sure need more botanists, more scientists, more people that care about the environment. So thank you all for being here and listening to me. Also contact me if you have other questions or ideas about things. Please reach out to me through email and Twitter. Yeah.
We do have one more question that came in that I'd love to ask. So it says, birders have a life list of birds they want to see. Do you have a life list of plants that you want to see and haven't yet?
That's a great question. Yeah, plant botanists, we really don't have a life list, but I'm trying to think of what plants I want to see. The baobab trees in Africa, I haven't gotten to see those. So that's a plant species I would love to see. I'm trying to think what else. Oh, well, witchia. It's like it's a beautiful plant. That was something I would like to see. So these are a couple plant species that I've seen in like botanical gardens, but I want to see them in their natural habitat. So yes, there are a couple plant species I would like to go travel to see.
Yes, and we hope that you get there, because I know COVID is making it so excruciatingly painful to be a field researcher right now.
Still haven't been to Australia yet, but my fingers are crossed that I'll get there soon.
Yes, and we are hoping that for you as well. I will go ahead, Dr. Williams, and post all that information that you gave me about Plants Are Cool Too, about Black and Botany Week, all these resources, because they're really great. So we will post that on the Asheville Museum of Science Facebook page. And yeah, we appreciate you so much spending your morning with us.
Yeah, this was so much fun. Thank you all for all of the great questions.
All right, you have a good afternoon. Thank you.
Thank you.
Bye.
Bye.